Hi everyone, my name is Shima. I'm a master's student at Universitat de Montréal under supervision of Dr. Blake. I'm involved in the vision team of courtois.neuromod project and today I'm gonna explain my master project which is about optimizing a graph convolutional network model for individual brain decoding using HCP-trt dataset. I will start today's talk with a brief introduction of brain decoding and the recent key works in this field. After that, we will have a quick review of GSIM project and I will explain that how we want to optimize it for using at the level of individual. Also, I'll give a summary about HCP retest dataset task and experiment design. And since so far I have trained a benchmark model on this dataset, at the end, I will show some of the benchmark model results as well. So let's start with brain decoding. One of the most discussed topic in neuroscience research is understanding human cognition. Basically, brain decoding relies on the fact that correlation can be established between a brain activity and the outside world. The main goal here is inferring the mental state and cognition using the non-invasive measures such as functional MRI scans. In this slide, you can see a schematic view of general brain decoding process. Analyzing brain activity pattern can reveal information such as what a person is seeing, feeling, or hearing, and so on. And as you can see in the figure, for example, we have fMRI scans of a few conditions, and the decoder will find a unique pattern associated with each of those states. Mainly, there exist two kind of approaches to tackle this topic. The first one is based on traditional statistical analysis and a general linear model using bold signals. And the second approach is based on using machine learning and deep learning techniques, which consider brain decoding as a classification problem and use classifiers to distinguish different conditions. Brain decoding using brain imaging data progressed significantly over the last decade. For instance, researchers have successfully used activation pattern for movie reconstruction or decoding the semantic context from words, also decoding visual stimulus. Even it is moving away from well-controlled experimental conditions to decoding fluid mental process such as dreams or intentions. And in terms of brain network in neuroimaging data analysis, there exists three possible definitions. As you can see in the figure, those definitions include spatial, temporal, and finally topological scales, which is ranging from individual nodes to the whole brain networks. Currently, artificial intelligence is trying to imitate the human brain activity, and deep learning techniques are able to do tasks that was previously impossible. Here we have an experiment of a Japanese team that was done on 2019. In this figure, we can see the procedure of the advanced deep image reconstruction for processing natural images. The link between image features and brain activity is found by using a linear regression model. And here the pixel values of the input image were optimized so the deep neural network features of the image were pretty similar to those decoded from fMRI activity. And the deep generator to produce natural looking image used those optimized pixels. The second figure is another experiment from the same team, which was done on 2020. But this time, they focus on the brain activity during attention. During an attention phase, subjects were asked to attend to one of the two superimposed images and ignore the other one. And the second part of the figure shows the reconstruction procedure, which is similar to their previous approach. They analyze the fMRI data using the deep image reconstruction model, and where they optimize the pixel values of an input to make it closer to the target features. But as you can see, the reconstructed images of both mentioned approaches are still blurred, and it mostly happened because of using a linear regression in the process. So the question is it really possible to generate a DNN's model to analyze brain activity directly without using any linear model? Also, I want to show another study of the mentioned team. In this study, they proposed the term brain hierarchy score called as a BH as a metric to quantify the degree of hierarchical correspondence based on the decoding of individual DNA unit activations from human brain activity. In this figure, they plot the BH scores of 29 different methods. By comparing these models, they found that simpler DNNs like AlexNet or VGG family have relatively higher scores, but models with high image recognition performance, such as the Inception family, show lower BH scores. And it means that recently developed high performance DNNs are not necessarily brain like and mostly are negatively correlated with image recognition performance. To talk briefly about previous model in terms of prediction brain cognitive states, I can say the previous approaches were generally shared common limitation, such as using a small data set that doesn't investigate the model generalizability or making a large collection using meta-analytic approach or even building a linear classifier using contrast map that neglect the temporal dynamic of cognitive process. And if they used Human Connectome Project dataset, which is a huge dataset, they were focusing on a single cognitive domain at a time, or maybe distinguish 7 cognitive domains. And so far, I can say few works have attempted to train a multi-domain brain decoding model that generalize across many different cognitive tasks from multiple cognitive domains. Also, the thing that which is important in my project is they didn't focus on individual brain decoding, which is a main goal of my project, as I told. The mentioned reasons was a motivation behind a GCN model generated by Dr. Zhang, and which I'm gonna present it briefly now. First, I'm gonna start with Human Connectome Project, which is a dataset that was used for this experiment. To develop a deep neural network, we need a large dataset, so HCP was chosen. And HCP is a huge multimodal dataset with over 1,200 subjects that contains a structural, diffusion, functional MRI, and also MEG. It has about 1 hour of functional data per subject when they were performing many different tasks from 7 different cognitive domains, which were working memory, motor, language, social cognition, relational processing, emotion, also gambling. Uh, In this experiment, gambling task was excluded because the experimental design of this event is too short, and that's why Dr. Jan and Dr. Blake decided to remove it from this experiment. And at the time for single fMRI volume is 0.72 with 2 millimeter ISO resolution. The organization of brain networks has been recently studied with graph theory. Graph signal processing provides a network representation of a brain organization and function by associating nodes to brain regions and defining the edge via anatomical or functional connections. Also, it can be used to transfer the functional imaging data into spectral domains. Basically, it provides a nonlinear embedding tool to project brain activities onto a Laplacian eigenspace that integrate spatiotemporal neural dynamics among connected brain regions and networks. The first step for building brain graphs is loading brain connectome. Second step is building a graph based k-nearest neighbor using the connectivity matrix. In this case, each node is connected only to k other neighbors based on connectivity strengths. And finally, the third step is making a Laplacian matrix that can be calculated from either adjacent matrix or weight matrix. As mentioned before, the GCN model is providing a network representation of brain organization by modeling the variety of brain dynamics on brain graph. This figure shows the pipeline of this model. First, the multimodal cortical parcellation was used to define the brain graph nodes, and resting state functional connectivity indicated the weights on the edge. Then, by connecting each brain region only to its 8 neighbors with the highest connectivity, the k-nearest neighbor graph was built. The model consists of 6 graph convolutional layers with 32 graph filters at each layer, followed by a global average pooling layer and 2 fully connected layers. And at the end, softmax was used to output prediction. Generally, this model takes a short time window of fMRI time series as input and then maps the fMRI data onto the brain graph. Then it propagates brain dynamics information on interconnected brain regions and networks and generates task-specific representations of recorded brain activities to predict the corresponding task label. This study results of 21 experiments were explored, and these 21 experiment conditions were explored with different time windows. This matrix shows the results of a GCN model running on 10-second windows on 1060 subject, and 10-fold cross-validation with shuffle spread were applied. As you can see, the confusion matrix showed a nice block diagonal, which means that the majority of the cognitive tasks were accurately identified. And even when mistakes happened, they happened for the cognitive states that are pretty similar, such as relational tasks. The average result for 10-second windows was around 90%, where the linear support vector machine returns just a 63% accuracy result. And the chance level was less than 5%, it was 4.8%. In this figure, the impact of the duration of fMRI time windows on cognitive annotation is demonstrated. Basically, the larger ephemeral time windows duration has the higher prediction accuracy. At around 6 second window, we reach at the accuracy over 80%, while for a single volume, which is in this case 0.72 second, the accuracy is still feasible. The other key question in this experiment was to figure out that how much data is necessary to recognize brain states uh, with an artificial neural network. Here we have a graph that shows the average prediction accuracy of these 21 states as a function of the number of subjects that were considered for the model prediction. As about 70 subjects, the model starts to return very accurate prediction, and the plateau is happened at around 280 subjects. Considering the 1 hour of functional data per subject in HCP dataset, it means that we need around 280 hours of data to train such a network. Let's move on to my project. My project main goal is investigating the performance of the GCN model for individual brain decoding using multiple tasks of the HCP-trt dataset. We hope to improve the results of the current model at the individual level by adding some modifications, such as fine-tuning for better training at the individual level. Also, in order to reduce the high dimensionality of fMRI data, rather than the statistic functional parcellation, which neglect the dynamic reconfiguration of brain network, the dynamic states of parcellation will be used. It means that instead of having graph nodes that are not task specific, we will use the state stability maps of each task. We hope the meaningful connection of looking at the individual data along with individual functional embedding helps to have a better results. The benchmark for HCP-trt dataset was investigating the results at the individual level using easy decoding tasks. The primary goal of this project was extending the research in this field and giving the opportunity to researchers to estimate how expandable is this science and how far we can push it to answer the scientific questions. Dataset consisted of 6 healthy subjects, 3 women and 3 men, with the age range between 32 to 49. And we aim to characterize those 6 brains in depth for each particular task. Also, the repetition time is a 1.49 with 2 millimeter is a resolution in fMRI data acquisition. Overall, for each subject, we have over 8 hours of functional data, which is way more than any other public data sets in this field. So it's open-ended, and there are a number of different experiments that can be done using this data set. For HCP-trt, the classical functional localizers developed by the Human Connectome project were repeated 15 times, and that's why it's named test-retest. The data acquisition happened during 10 sessions. In each session, either 2 repetition of 7 HCP tasks or 1 resting state and 1 time of 7 HCP tasks happened. In this table, we can see more details such as number of condition and volume per task. Generally, these 15 times repetition enable us to check if we can accurately characterize those cognition states for each individual. For my project benchmark model, first I applied a post-process stage to data to generate a condition-specific beta maps. Using the GLM model with 5 millimeter Gaussian kernel smoothing, and also params 9 library was used for loading confounds. And the evaluation was done for just a single cognitive domain at a time, using the beta maps regression that was generated during mentioned post-process step. The classifier is a naive linear support vector machine with five-fold cross-validation and a test size of 20%. This plot shows the average prediction results for all the six subjects for motor and working memory tests. Motor was chosen because it's the simplest, and the working memory was selected because it's one of the most challenging tests. The results are very promising considering the fact that a simple linear classifier without any feature selection was trained on these subjects. For example, we have 99 or 98 for motor tasks that are really high for this naive classifier. Um, these two figures show the previous plot with more details and all the subtasks. We see that most of the subjects follow the same trend and show respectively higher or lower prediction result for different tasks. And here as an example, we have the weight maps beside the activation maps for a same subject. And in this case, the motor task was chosen. And as you can see, both maps shows the quite similar pattern that anatomically makes sense as well. And in this slide to show how maps are different between high prediction and low prediction results, um, I put the maps of motor tests for two different individuals, more specifically the one who returns 98 accuracy and the other with the lowest accuracy result, which was 65%. And we see how this activation maps dispersal affect the prediction results. For the subject that the activation placed in anatomical meaningful area, we have significantly high result, while for the other one, activation is scattered. Finally, in terms of conclusion, I can say since GCN is a network that can be applied to any kind of data set, and in the previous work, as you saw, not only it recognized brain conditions, but also it had to learn brain variability, which is really considerable from one subject to another. So we aim to train the network at the level of individual to see how much we can cut down this data requirement. My project next steps would be experimenting the effect of the reaction time and human-like behavioral characteristic on decoding results. Also, we will check the prediction result on a smaller time window, and finally, we will run and optimize the GCN model on HCP-trt using the dynamic states of parcellation, which is the last step of my project and final goal. That was all my talk for today, and I would like to take a moment to thank Neuromod teams, especially Dr. Blake, my supervisor, and Courtois Foundation. And thank you all for your attention and listening.